The AAAS Newcomb Cleveland Prize, our oldest award, recognizes an outstanding original research paper published in Science each year. The winning paper is chosen based on the quality of its scholarship, innovation, presentation, likelihood of influencing its field, and its wider interdisciplinary significance. This year, we present the prize to the paper entitled Hunting the Ego Killer, a cyanobacterial neurotoxin causes vascular myelopathy, first published March 26, 2021. The paper was the product of an international collaboration co-led by Susan Wild, Associate Professor of Aquatic Science at the University of Georgia, and Timo Niedermeyer, Professor of Pharmacognosy at Martin Luther University Halle-Wittenberg in Germany. At DeGray Lake, Arkansas, in fall 1994, a mass die-off of bald eagles initiated a wildlife investigation. Dozens of birds died, and many displayed odd behavior, like missing their perches and flying into rock walls. When wildlife biologists examined dead eagles, they found extensive lesions throughout their brains and spinal cords. By 1998, scientists knew that birds from at least 10 different southeastern reservoirs in 5 states had this new disease called vascular myelopathy, or VM, but no one knew the cause. In addition to avian species, Susan Wild and UGA colleagues documented that VM could affect amphibians, reptiles, and fish. Researchers made gradual progress uncovering the cause. Wild and her colleagues in South Carolina, where many of the birds suffered from VM, found dense growth of the invasive plant Hydrilla verticillata. The plant, imported from India for use in aquariums, was overrunning man-made lakes in the southeast, readily consumed by coots, which are frequently eaten by bald eagles. Using fluorescence microscopy, Wild observed that in affected water bodies, hydrilla leaves were densely covered with cyanobacteria colonies. This novel cyanobacterium was also found in all the reservoirs where the birds had died. Wild and her colleagues speculated that the cyanobacterium produced a toxin responsible for the eagle deaths. They fed plants collected from affected water bodies to laboratory birds, confirming the plants colonized by cyanobacteria caused VM. The cyanobacterium was named Autochtonos hydrolicola, Greek for ego killer living on hydrilla. However, no known toxin was detected in the cyanobacterium. In Germany, Niedermeyer and co-authors grew this cyanobacterium in the lab. However, lab-cultured bacteria did not cause VM in test chickens. Hypothesizing that the toxin might be produced in lakes but not in the lab, they collected more hydrilla leaves from impacted lakes. Direct analysis of the cyanobacteria growing on these leaves revealed a compound not present in lab-grown autochtonos that contained 5 bromine atoms. The team was able to reproduce the compound in the lab when the cyanobacteria were grown in the presence of bromide. The compound was revealed to be a novel biindole alkaloid, and further experiments confirmed it causes VM. The scientists called the compound autochtonotoxin, toxin that kills the eagle. Jana Marish at the Biology Center of the Czech Academy of Sciences led the genome sequencing of Autochtonos hydrolicola, which allowed the identification of the compound's biosynthetic gene cluster. Bromide can leach from rocks, but it can also be introduced into the environment from human activities such as coal-fueled power plants or water treatment facilities. The authors recommended increasing monitoring and public awareness campaigns be implemented for hydrilla and autochtonotoxin to protect both wildlife and human health. The study brought researchers together from multiple countries and disciplines to solve a decades-old mystery, and reminds us how dangerous human-induced changes to ecosystems can be. These unanticipated effects are only likely to increase as we continue to alter the world around us. Hunting the ego killer, a cyanobacterial neurotoxin causes vascular myelopathy, recipient of this year's AAAS Newcomb Cleveland Prize.